Good morning, Shailesh sir.
Good morning, madam.
So, good morning.
Good morning, sir.
Good morning. Good morning.
मेरे मेरे को फोटो दिख रही है ना मेरी? ठीक। Dr. Geeta, start.
Good morning, everyone.
I'm audible.
Yes, sir, you're audible, sir.
Good morning, everyone. I'm Dr. Priyanka Kriplani, and it's my pleasure to welcome you all for the National Conference on Advancing Pharmaceutical Research and Methodologies in Innovative Drug Delivery, Phytopharmaceuticals, and Healthcare Policy Integration, organized by Guru Gobind Singh College of Pharmacy, Yamunanagar, and Amar Shahid Baba Ajit Singh Jujar Singh Memorial College, Bela, in collaboration with Association of Registered Pharmacists, which was established to promote the interest of registered pharmacists and to advance the profession of pharmacy in the country. Guru Gobind Singh College of Pharmacy, it was established in 2002 with a vision to be center of excellence of international repute in pharmaceutical education and research, and it is the first college in Haryana to be accredited by NBA and NAC. Amar Shahid Baba Ajit Singh Jujar Singh Memorial College, Bela, it was established in 1993 with a vision to promote quality education and training to students to make them able to face emerging challenges of globalized technology, competitiveness, and entrepreneurship. It is NBA accredited and ranked 57th in NRF in 2024 and also recognized in AICT Platinum category in 2022. Now, moving on towards the conference aims to explore cutting edge methodologies in pharmaceutical research, focusing on innovative drug delivery systems, the potential of phytopharmaceuticals, and their integration into healthcare policy. It seeks to foster interdisciplinary collaboration among the researchers, practitioners, and policymakers to address contemporary healthcare challenges. Today, it's our honor to have among us Dr. Zabir Ahmed, Director, CSIR, Indian Institute of Integrative Medicine, Jammu, as our chief guest of the day. Dr. Deependra Singh, Chairman, Education Regulations Committee, PCI, New Delhi, as our guest of honor of the day. Dr. Indu Kaur, Professor, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Dr. Geer M. Ish, senior faculty member at the Department of Pharmaceutical Sciences, University of Kashmir. Dr. Ashish Baldi, Professor, Department of Pharmaceutical Sciences and Technology, MRS Punjab Technical University, Bhatinda, Punjab. Dr. Sudhir Bhardwaj, Director, Divine International College of Pharmacy, Gwalior, Madhya Pradesh. Dr. Kumar Gaurav, Principal, Guru Gobind Singh College of Pharmacy, Yamunanagar. Dr. Shailesh Sharma, Principal, Amar Shahid Baba Ajit Singh Jujar Singh Memorial College, Bela. Welcome all of you. Now I request our Principal, Sir, Dr. Kumar Gaurav, Sir, to please welcome our guest. Please, Sir.
Thank you. Good morning, esteemed guests, distinguished speakers, and participants. As the principal of Guru Gobind Singh College of Pharmacy, Yamunanagar, it is my great privilege and honor to welcome you all to the National Conference on Advancing Pharmaceutical Research, Methodology in Innovative Drug Delivery, Phytopharmaceuticals, and Healthcare Policy Integration. This prestigious event is jointly organized by Guru Gobind Singh College of Pharmacy, Yamunanagar, and Amar Shahid Baba Ajit Singh Jujar Singh Memorial College of Pharmacy, Bela, and in association with the Associated of Registered Pharmacists. Today, we are fortunate to have among us some of the most eminent personalities whose achievements and contribution to pharmaceutical sciences and education inspire and illuminate our path forward. It is with immense pride that I welcome our chief guest, Dr. Zabir Ahmed, sir. Welcome, sir. Director of CSIR, Indian Institute of Integrative Medicine, Jammu. Dr. Ahmed is a trailblazer in pharmaceutical sciences, renowned for his groundbreaking research in metabolic disorders and inflammatory disease, sir. Sir, your dedication to advancing therapeutic sciences serve as a beacon of hope and innovation for the pharmaceutical community. I welcome you, sir. I extend a warm welcome to our guest of honor, Dr. Deependra Singh, Chairman of the Education Regulation Committee, Pharmacy Council of India, New Delhi. Sir, your steadfast commitment to shaping pharmaceutical education and equipping to the next generation of pharmacists with the skills to meet the ever-evolving challenges of healthcare is truly commendable, sir. And we are privileged to have with us Dr. Indu Kaur, professor at Punjab University, who is fondly known as the patent queen of India. with over 22 patents and recognition among the top 2% of pharmaceutical scientists globally. Ma'am, your visionary work exemplify how passion and perseverance can redefine boundaries. Thank you very much, ma'am, and welcome to our conference. A heartfelt welcome to Dr. Ashish Baldi, Professor at Maharaja Ranjit Singh Punjab Technical University, Batinda, and recipient of National Teacher Award. Sir, your transformative contribution as a mentor and educator highlight the power of teaching in shaping lives and future. Thank you very much, sir. I also extend my gratitude to Dr. M. Ishaq, sir, professor at the University of Kashmir, whose academic brilliance and unwavering dedication to research have left an inedible mark on the world of education and knowledge, sir. Thank you very much, sir. I also welcome you again as we embark on this journey. Today, let us draw inspiration from the remarkable contribution of these exceptional individuals and strive to create a future defined by excellence and innovation. Thank you for joining us, and I wish you an enriching and memorable day ahead. Thank you very much, sir.
Thank you very much, sir. Now I take the privilege to welcome our distinguished guest of honor, Dr. Deependra Singh, Chairman, Education Regulations Committee, PCI, New Delhi. He is presently working at University Institute of Pharmacy, Pandit Ravi Shankar Shukla University, Raipur. He has around 19 years of teaching and research experience. His prime research areas include formulation of multiple bioactive delivery systems for wound management, designing strategies for protein and peptide delivery, advanced statistical and mathematical modeling methods, tissue culture techniques, and designing novel systems for drug delivery of plant actives and extracts. He has published more than 110 research and review papers in national and international journals, with H index of 30 and I-10 index of 90, with citations of more than 4723. He has two Indian and two international patents granted and has completed several projects funded by UGC, AICT, ISMR, and DST. He is worldwide in the field of pharmacy and pharmacology, surveyed by Stanford University in the year 2021, 22, and 23. He is recipient of many prestigious awards, and recently he has been conferred with the prestigious APTI Dr. LVJ Nargund of Nargund Research Foundations for the Professor C.J. Shishu Award for Research in Pharmaceutical Sciences, a recognition that highlights his exceptional teaching abilities, research accomplishments, and dedications to shaping the future of pharmacy education. I welcome you, sir, and I request you, sir, to please address the audience,
Thank
please,
you.
sir.
Thank you, Priyanka. Thank you so much for introducing me. And indeed a privilege to among the stars of pharmacy and learned speakers who are present here. Honorable Chief Guest of today's function, Dr. Zabir Ahmed, Director, CSIR Indian Institute of Integrative Medicine, Jammu. Eminent speakers, my personal friend, Dr. Ashish Baldi, he is there, Professor, Department of Pharmaceutical Sciences, Indu ma'am, well-known mentor and researcher. Our teacher, Dr. Geer sir, who is present here, and as for my knowledge, these are the learned speakers of today's function. First of all, I'm very thankful to my personal friend, Dr. Kumar, who is always there, and indeed he never asked me for date. He instruct me, you have to present at that day. So thank you for having faith in me and for inviting me in this conference. Talking about your topic of the conference, indeed, it is always a very hot topic when we talk about integrating the natural products with the advanced delivery systems. And also one more commendable thing, what I think, what I can see, that organizing a program with integration of two institutions and including a pharmacist or an association of pharmacists, again, it is a good job, because we know that there are two competitive colleges, never they come together for anything, but two leading colleges of different states having a very good vision and collaborating and integrating for a purpose, for a cause, for organizing this national conference. So many congratulations to all of you for taking this approach. And if you'll see, present time, if you'll see that it is a very, I always used to say this is a changing time, fastly changing every day. If you'll see, you'll find change, because now the half-life of idea and half-life of technology, it is very less. Maybe you're working today on a particular strategy or a topic, you are talking about that, and similarly at the same or within a few days you will find that something new is there. So the need, what it is need of our, that is taking pace and keeping pace with the what it is changing time with the time. So always this is a responsibility on the scientists and leaders to nurture their coming generation, to intimate, to introduce them that what the challenges are coming. So I'm very happy that you have taken this topic to integrating, and also integrating this approaches or what we are taking with the policies, those who are involved in the policy making. So my congratulations to all of you, and I'm sure that what the deliberation you are going to have in this conference, it is going to contribute to the new researchers and the people who are working in this area. And the natural products are always very lucrative, but one bitter truth is also there, that we have to do good in that area. We are a country who is having a long history of natural products, having a therapy based on natural products. Ayurveda is well established therapy, but at the same time we are also facing problem when we are talking about the export of our material. We are talking about establishing the credentials of our therapies. So a lot of responsibility is there on the shoulders of the people who are working in this area. And but in the recent time we are doing very good. We are leading the world in these areas, and people are relying on us. And now if you see the strategy of government of India, they also given the full-fledged recognition to the people who are practicing in this area. So now the responsibility becomes much more than what it was previous, because now we are full-fledged practitioners. You have been recognized in the equivalent to the other practitioners who are doing practice in the medicine. So again, it becomes our responsibility that we should do very good in this area. I'm very hopeful that the today's conference, the deliberations will be very fruitful, and definitely the people will be benefited by the deliberations of today's conference and the various speakers who are invited here. So thank you so much for inviting me this function, and my best wishes to the conference. It will be a very successful conference. Thank you so much. Jai Hind, Jai Bharat.
Thank you very much, sir, for your kind words and most importantly, your time. Thank you very much, sir. Now, I take the privilege to introduce our distinguished chief guest of the day, Dr. Zabir Ahmed, Director, CSIR, Indian Institute of Integrative Medicine, Jammu. Dr. Ahmed is heading CSIR, Indian Institute of Integrative Medicine, a premier lab of Council of Scientific and Industrial Research under the Union Ministry of Science and Technology, Government of India. He is actively pursuing research on rheumatoid arthritis, inflammation disorders, diabetes mellitus, and obesity, including the ambitious research project on discovery of cannabinoids and their derivatives. He is also leading several societal
Voice is breaking.
I think so. Voice is not coming. Dr. Kumar, can you hear me?
Voice is breaking.
Yes,
Sir, I think there is a technical glitch.
Okay, please.
Priyanka ma'am, please continue.
He is recipient of prestigious Knowledge Partnership Award of CSIR and JNK-UT Award for Excellence in Science and Technology for significant contribution towards science and society. I welcome you, sir, and I request you, sir, please address the audience. Please, sir.
Thank you, Dr. Priyanka, for giving a nice introduction for which I think I am not deserving. For a moment, I was confused that you were mentioning about me only. So thank you very much. And sincere thanks to guest of honor. I think he has done my job. So other dignitaries, Dr. Ashish Baldi, Dr. Indu Kaur, Dr. Ishaq, and Principal of Guru Gobind Singh College of Pharmacy, Director of Other Coordinating College of Pharmacy, delegates from CSIR Triplement Jammu and from Jammu and Kashmir, a warm good morning to everybody. Indeed, this is great opportunity for me to be a part of this national conference on a very important theme. Like Dr. Deependra mentioned, we have rich nature resources. If we talk about Jammu and Kashmir, we talk about northeastern state, we talk about Ladakh, huge natural wealth is there. And at the same time, if we consider our traditional system of medicines, we are carrying very rich legacy. The requirement is that how we can just globalize our traditional knowledge. And here the requirement of such deliberation is there. And what I see, the organizers, the other team members who are taking such initiatives, these initiatives are not having academic importance, but what I feel, the kind of topics which will be deliberated during this conference, these topics have national impact, international impact. So if we talk about the integration of our traditional medicines with modern medicines, active research, active standardization, active quality control of such traditional knowledge and traditional medicine is required. And this is the wonderful job the two institutions are doing. On this occasion, from my side, from my council side, I take this opportunity to congratulate all the organizers, and I express my sincere thanks to all the organizers, Dr. Geeta, Dr. Kumar, Dr. Sharma, for making me part of this wonderful event. I will not take much of your time. Just I would like to share few slides so that one topic there in this theme is phytopharmaceutical. This institution, CSR IIIM, is expert in this, and I would like to share the kind of work this council, this institute is doing as far as phytopharmaceutical drug discovery program is concerned. So just I will take this opportunity to share my screen. Then actually, normally we use MS Teams. Zoom for the first time we are using in this system. I will check if some setting is required. My screen is visible or not? No.
No, sir.
Oh, okay. Just. Sorry, give me 1 minute, please. Yeah.
Okay.
So is it visible?
Yes, sir.
Screen
Yes,
is- Sorry, I have just put everybody.
issues.
on hold. So just I take this opportunity to just introduce the kind of work we are doing in this phytopharmaceutical area. Before that, I would like to just give brief about the CSIR and the institute and its history. So CSIR, we are having all over India, we are having 38 institutions. All these institutions, as per their domain areas, research areas, they are divided into different clusters. Like the biological sciences labs are in a biological cluster, then chemical sciences cluster is there, engineering sciences cluster is there, information sciences, physical sciences, and beside this we have many extension center all over India. And recently in Mumbai, CSIR Innovation Complex has been inaugurated by Honorable Union Minister of Science and Technology. In the council, our Honorable Prime Minister is President of CSIR, Honorable Union Minister of Science and Technology is Vice President, and Director General presently Dr. N. Clay Silvi is heading all these 38 institutions of CSIR. So about CSIR IIIM Jammu, we are having two centers here. One is in Jammu and other is in Srinagar. So if we talk about the history of this institution, since the region, like I talked a little earlier, that we have rich nature sources are there right from Ladakh to Jammu and Kashmir and other parts of North India. So initially, when it was started in 1941, the center was focused in the development of nature products from herbal resources. We were used to just do chemical characterization, isolation of molecules, then pharmacological activities were used to be performed. And Colonel R.N. Chopra, who was son of Soil, so he founded this institution in 1941, well before the independence, our independence. So later on, it was renamed as Regional Research Laboratory, and it was taken over by Council of Scientific and Industrial Research. And present day, it is Indian Institute of Integrative Medicine. When we talk about the integration of traditional medicine with the modern medicines, the institution like CSIR IIIM, they are playing active role as far as standardization of these traditional medicines is concerned, quality control of these medicines is concerned. So this institution has pivotal role in the integration and modernization of our traditional medicines. So, like I talk about the kind of rich nature resources, we have different experimental farms all over Jammu and Kashmir, and in Ladakh we have recently acquired one farm where we are doing medicinal aromatic plants cultivation there. So, little bit about the kind of farms resources we have. So if we talk about the preclinical drug discovery, this institution, CSIR IIIM Jammu, is a composite model for preclinical drug discovery. In the region, we have natural products, we have natural resources. Beside this, an important component, that is microbial species. If you talk about the hot spring in Ladakh and other natural resources are there where microbial biodiversity is there. So like I mentioned, this institution is a composite model for preclinical drug discovery. So we are following end-to-end approach as far as drug discovery program from nature resources is concerned. So if we see the globalization and if we see the global conditions and global scenario of the drug discovery, on one side we have traditional system of medicines, we have nature resources are there. So what I feel, the kind of nature sources, the biodiversity is available, infrastructure is available, and our endeavor is to place our traditional medicines, phytopharmaceutical into global market, and this kind of approach we are following in this institution. Little bit about the kind of facilities we are having as far as preclinical drug discovery is concerned, right from GLP standard animal hosts, where we used to do the regulatory toxicological studies. We have state-of-art fermentation facility, where upscaling, where the API synthesis upscaling is done. Then we have BSL-3 facility. Beside this, all kind of facilities which are critically required for standardization, bioprospection, pharmacological evaluation, then the regulatory toxicological studies of these leads. So all kinds of facilities and infrastructure are available in IIIM Jammu. So we have bioprospection unit where the researchers are involved in exploration and bioprospection of natural resources from Jammu and Kashmir, Ladakh, and other parts of the country. And in addition to this, follow-up in the line of drug discovery, we have well-established natural product discovery group where the exploration, characterization, isolation of extracts, isolation of pure molecules is done by the chemical sciences group. Then we come to the pharmacological division of this institution, where we are working in different disease area, and our main focus is to evaluate certain NCs, to explore phytopharmaceuticals, to develop phytopharmaceuticals as drugs for various diseases. And beside this, another area we are involved in, that is nutraceutical also. So like I mentioned, if we see, presently India is looking towards West, India is looking towards China for import of APIs, for imports of many other drugs. But back home in India, we can see we are bestowed with rich traditional medicines. We have Ayush system of medicine is there. We have allopathy system of medicine is there. So only the requirement is that, like all stakeholders, whether industry people, whether institution like your institutions, government organizations, their efforts are required to explore the gold mine we are sitting on. And in line with the development and advancement of the pharmaceuticals, phytopharmaceuticals, so another class of drug is there, that is phytopharmaceutical. So I will give a little bit about the phytopharmaceutical drug discovery program. So if we talk about the Indian system of medicines, you all are well aware, like the traditional system of medicines there, which is linked with the social fabrics and the lifestyle and the cultural linkages there. Rich pathies are there, but the standardization, like I talked about, the standardization, the quality control, the proof of concept to these traditional medicines is required, and we are just putting our efforts to do modernization of the traditional medicines. Here, the key questions are, when the phytopharmaceutical guidelines came into force, the key questions are how we can put our traditional knowledge, ancient knowledge, so that these the traditional medicines, these traditional products are accepted globally. And another, like I mentioned, if we are talking about APIs, we are talking about other critical medicines, so we are looking towards rest of the world. So here the policymakers, they felt that our efforts should be like, we should globalize our traditional knowledge, we should globalize our local knowledge. At the same time, we should localize the global knowledge. So what I feel, like the phytopharmaceutical drug development was adopted in India. So it is the main objective of this bringing this act is to standardize our traditional medicines, to do chemical characterization, to do captive cultivations of these traditional plants, so that we will be able to place our natural resources into Indian market, into the global market. So here the act came in 2015. This Phytopharmaceutical Act was implemented and enacted, and if we talk about what is the phytopharmaceutical drug, so if we talk about the traditional system, Ayurveda formulations are there, Siddha formulations are there. But with the passage of time, we need to run with the time. We need to follow the time. Like if we want to put these traditional medicine or medicinal plants products to advanced market into the global market, so some standardization is there. In this act, it was defined that any standardized plant extract, it may be a part of the plant, but it should have 4 bioactive or normal phytochemical compounds. We have allopathic system of medicines. The guidelines for drug discovery is there, but at the same time, parallel to allopathic system, these phytopharmaceutical guidelines are there, where we are doing exploration and we are doing research. It is similar to the kind of research, kind of preclinical drug discovery we are following in allopathic system of medicines. So if we talk about the route identified for phytopharmaceutical, that is only for oral route. It should not be through parental route. So I will just skip this slide. If we talk about the requirement in the global market and our share in global market, we have rich resources, but at the same time, if we see the percentage, we are only contributing 1 to 2% as far as requirement of herbal products in global market is concerned. So regulations are there, our research labs, these are not only CSR lab, other industry, other people are also involved in phytopharmaceutical drug discovery program. So the kind of studies we required to do in phytopharmaceutical, I think all the experts and dignitaries, they know about the kind of studies. If some delegates are there for the information of these students and delegates, if we talk about the preclinical drug discovery phase, right from standardization, like I mentioned, chemical standardization of drug or the plant product with 4 chemical markers has to be done, then preclinical efficacy, in vitro, in vivo, then a couple of tox studies, acute oral toxicity in 2 species, then dose range finding studies, then subacute toxicity studies are required to be done for phytopharmaceutical products. Then PAPI standardization, followed by stability studies and compilation of doser and filing of IND doser for clinical trial. So here in CSIR phytopharmaceutical mission, our main focus is 2. Like if we talk about traditional system of medicines, we have herbal mineral formulations are there, we have herbal formulations. But if we talk about, and if we are concerned about the quality, the standardization, the proof of concept, scientific proof of concept to these formulations, that is missing. On the basis of that, so in CSIR phytopharmaceutical mission, our main objective is the sourcing of the plant material, captive cultivation of these herbal resources, plants materials, processing, quality control, value additions, then followed by the exploratory studies for certain kind of disease targets. So this is the glimpse of the captive cultivation we adopted in phase 1 and phase 2 of phytopharmaceutical all over India. Many labs are involved. Besides CSIR Triple IM, other labs are also involved as per their expertise. So CSIR Triple IM Jammu is a nodal lab for phytopharmaceutical mission in India. So little bit about the kind of progress we made in phase 2 and phase 3. In phase 2, we identified 2 formulations. We did PAPI preparation, stability studies were completed, pre-clinical efficacy, toxicological studies were completed, and in phase 3, we are putting these formulations for clinical trial, like INDs for these formulations have already been filed. So beside this, another important area we are considering, that is the cannabis research in the cannabis sativa. This plant is prohibited plant. CSIR-IIIM Jammu is first in government sector. We were given this license by Jammu and Kashmir government for completing the basic preclinical research, and we have completed. So again, this is another phytopharmaceutical entity we are working on. So this gives the glimpse of the involvement of various labs all over CSIR. CSIR-IIIM is nodal. Various disease targets are there in phase 3 of phytopharmaceutical. So two main verticals we are focusing in phase 3 of phytopharmaceutical. One is captive cultivation. Like I mentioned, when we talk about the standardization of any part or any extract of the plant, on the basis of the 4 markers, another important challenge is there, how to maintain the range of the specific identified 4 markers. So in this, we have identified that in one vertical, the captive cultivations of all shortlisted plants and the plants we are working on. So the captive cultivation should be done so that tomorrow we will be able to present and we will be able to demonstrate before the policymaker that such kind of ranges should be adopted as far as standardization of phytopharmaceutical drugs are concerned. So beside this, many plants are there and preclinical and clinical development being followed by different CSIR labs all over India. So just a glimpse of the phytopharmaceutical leads identified by CSIR-IIIM. I mentioned about Crocus sativus, that is for Alzheimer's disease. IND has been filed. It means all kind of preclinical studies we have completed. IND has been filed. There are 2 phyto leads for this rheumatoid arthritis. For one, IND has been filed, and for other IND enabling studies are being done. So beside this, Boswellia serrata and cannabis are other 2 drug leads for phytopharmaceutical drug discovery route. And beside this, a couple of NCs are there. Just to mention about this IIIM 290. So we are starting with the phase 1 and phase 2 clinical trial for this. And if all clinical trials will be completed successfully, this will be first molecule from India which will be used orally for management of pancreatic cancer. So these are few phytopharmaceutical leads where captive cultivation is being done by IIIM or other lab, IHBT and CMAP. So a couple of success stories so far, like I mentioned about Crocus. So IND has been filed and we are developing this product as phytopharmaceutical drug for management of Alzheimer's disease. So Farmenza Herbal is the company which is in collaboration with us, and collaboratively we are just developing this phytopharmaceutical for Alzheimer's disease. Like I mentioned, this rheumatoid arthritis, disoxylum is there. So IND has been filed for this, and we are in process of preparing some formulations, and we will be going ahead. Another lead for rheumatoid arthritis, that is Virginia ciliata. So a couple of IND enabling studies are left, and we will be completing this also. So like I mentioned about the cannabis is legalized in US, in some other countries, but in India it is prohibited. And if we see the kind of composition, the kind of phytoconstituents available in this plant, so this single plant has phytoconstituent for more than 5 diseases. So identifying the need of doing some research, doing some exploratory studies on this. So we applied to Jammu and Kashmir government. Jammu and Kashmir government has granted us to do some basic research. Our research group has completed all kind of preclinical studies as far as pancreatic, as far as cancer pain management is concerned. We identified one PAPI, which is potential for managing the pain in terminal cancer patients, and we are in collaboration with the Tata Memorial Hospital of Mumbai. And once the formulations license will be granted and transportation license will be granted, we will be moving ahead for clinical trials for this cannabis lead of phytopharmaceutical. So this is the team and the other achievements as far as phytopharmaceutical drug discovery program is concerned. I hope I have not taken your much of time, so I am within time. So thank you very much for giving me this opportunity. And what I see, the kind of initiatives these two pharmacy colleges have started, that is wonderful. It is not having any relevance as far as academics is concerned. We need to bring all industry people, all stakeholders, policy makers, researchers on board, and that is what I see through this conference. The organizers have tried in a wonderful way that all the stakeholders are there and they are just deliberating, and I look forward for a wonderful proceeding during a day ahead. So thank you very much. And finally, I will take this opportunity to invite all the dignitaries who are present here and all the delegates and the staff and the principals of these colleges to visit CSIR IIIM Jammu. And definitely we will be able to collaborate with you all people as far as if we talk about the sustainable development goal, Viksit Bharat, all our institutions should come together. And for that purpose, I take this opportunity to invite you people to CSIR IIIM Jammu. Once again, thank you very much. Thank you, organizers, for giving me this opportunity to be with you for this wonderful session. Thank you very much.
Thank you very much, sir. First of all, for sharing your valuable insights about the CSI at IIIM, about the various facilities and resources and the important researches that are going over there, and for your kind and encouraging words, and most importantly, your presence. Thank you very much, sir. I also like to extend a warm welcome to Dr. Peer Ghulam Nabi, who is CEO, Guru Nanak Khalsa Group of Educational Institutions. He has also joined the conference. I welcome you, sir.
Thank you, Dr.
Now,
Thank you.
yes.
Thank you, Dr. Priyanka. Thank you so much. I'm glad to see Dr. Geer here, Professor Geer from Kashmir, and Dr. Zabir. Thank you all for joining this wonderful conference. I'm sure all the participants would benefit from your wisdom, your scientific knowledge, and the researches you have contributed in your respective fields of pharmacy. And thank you all. I'm not from pharmacy background, but it was pleasing to listen to Professor Zabir's talk about how pharmacy research is relevant to the industry, as well as looking at the not only traditional knowledge that we need to focus on, but also looking at the globally recognizing institutions, how they have contributed to the medicine and addressing the challenges that we are facing and the challenges which we would be facing in future. And I look forward to attending all the sessions here. And thank you all, and congratulations to Dr. Gaurav and Dr. Geeta, Priyanka, and all the organizers here. Thank you.
Thank you very much, sir, for your encouraging words, and always be with us all the time. Thank you very much. Now, I would like to, now moving on towards the technical session, I would like to introduce our first speaker of the day, Dr. Indu Kaur ma'am. She is currently working as Professor of Pharmaceutics, UIPS, the DPIIT IPR Chairperson, Punjab University, Independent Director, Nectar Life Sciences Limited, and Secretary, the Indian Pharmaceutical Association Punjab branch. She is almost the top 2% most cited scientists across the globe as per the data promulgated by Stanford University, USA. Her research forte is enhancing bioperformance of drugs, plant extracts, phytochemicals, and biomolecules, siRNA, proteins, peptides, and probiotics using active tailored solid lipid nanoparticles, nanovesicles, and electrospun scaffolds as novel platform nanotechnologies. Emphasis of her work lies on industrial and clinical translation and is reflected through licensing of 4 of her technologies, 21 granted patents. She has received funding of around 100 million INR from government and industry And based on her contribution to the field of IBR, Professor Kaur was invited as a special guest to the Government of India on the 75th Republic Day by the Office of Controller General of Patents, Designs and Trademarks, Ministry of Commerce and Industry, DPIIT, Government of India. She recently featured as a persona of eminence on the Change Makers episode organized by AICT MOE Innovation Cell on the importance and role of patents in the innovation ecosystem of AICT. She is also a recipient of many prestigious awards. So, ma'am, I request you to please shed light on the topic Traditional Knowledge Meets Modern Science, a New Era of Phytopharmaceuticals. I welcome you, ma'am. I request you, ma'am, to please take over.
So good afternoon, everyone. I think we are still in the good morning zone. So thank you, Dr. Priyanka, for a very kind introduction. And I'm grateful to Dr. Kumar Gaurav and also Dr. Geeta for inviting me today. Respected Dr. Zabir Ahmed, who was the chief guest for today's program. And I just listened to him and the work which is being conducted at CSIR, IIM, Jammu on phytopharmaceuticals. That was very interesting. We also had Dr. Deependra Singh with us, who was heading the education regulations for PCI, and also Dr. Kumar Gaurav I mentioned, and Dr. Sailesh Sharma from Bela College. Hello, Hansi. And my co-speakers, Professor Ashish Baldi and Professor Geer Ishaq from University of Kashmir. So thank you so much for having invited me today. And without taking much time, I'll just try and move to my presentation. So can you see my slides?
Yes, ma'am.
Okay. So based on the theme for today's program, I also took up a topic where I would say that trying to, and that's my favorite thing is that moving these natural molecules or phytochemicals from the domain of just being curatives. अभी तक हम जो भी बात करते हैं उनके बारे में, we always say that you take them and they would protect you against the diseases. So it is my fervent wish that we move them from these protectives to curatives. And the only answer to that is by modern pharmaceutical technology. और मैं कहती हूँ अक्सर कि उसको एक pharmaceutical कुटौर दे दो, जैसे कि हम देखते हैं आजकल जो फैशन वॉक होता है उसमें उसको एक ऐसे कपड़े पहना दें हम ड्रग डिलीवरी के कि वो उसकी बायो परफॉर्मेंस एनहांस हो जाए. And that is what we can do together, coming together of the scientists, biochemists, and formulation scientists together, as well as people from traditional know-how. And because folkloric knowledge was developed based on observational data collected over hundreds of years. So वो काम तो करते हैं, लेकिन जब हम उसकी साइंस की बात करते हैं बड़ी बारी we do not have an answer to it, and at other times we see that when we try to bring them to clinical settings, they do not perform. So I'll just take you across my journey trying to bring these molecules further into the domain of therapeutics. So this is a brief about my institute, which is University Institute of Pharmaceutical Sciences. We are one of the oldest in the country. We were set up pre-independence in 1944, and we are very proud of having rich faculty like Professor Kishan Singh, who was the only Padam Shri in the area of pharmaceutical sciences, a well-established researcher, educationist, one drug coming out of UIPS. It is a very proud honor for a department in a state university. We have series of new molecules, technologies transferred to the industry, and there are 8 products in the market from the institute. So this is a brief about my lab, the type of equipments we have, and the motto we have, where we talk of versatility, simple technologies which are scalable, getting on to safe products which are stable and biocompatible. Because biocompatibility is a big concern when we talk of nano drug delivery systems. And this is a famous saying which we all know by Hippocrates, much before Christ, and it was said that let food be thy medicine. So I just want to expand it to, so maybe when he said food, he was talking about the plant products, the natural molecules as well. And as I said, my wish, or my vision is to move these molecules from being preventives to curatives by giving them a new kutor. So phytopharmaceuticals, already Dr. Zabir Ahmed has very, explained that these are, it is important that they are purified and standardized, and they are usually the rich fractions, and they need to have at least 4 markers which would define them. These could be quantitative or qualitative, and they are usually for internal as well as external use, except that they cannot be given parenterally, and they are not defined like this in the Ayurveda. So, and they fall into the domain of the drugs. So what are the challenges? Why can't we directly move these phytochemicals into the therapeutic armamentarium? The solubility. So most of these molecules, I don't know how many of you know of the term hormesis. And so these were basically developed within the plants, or are produced in the plants, to protect them against consumption by animals. So they exist within the plants, and they cannot be, so that the plants are not destroyed or eaten up. So at times they exist in forms which could be not very useful to the humans. So solubility is a big issue with most of these molecules. Stability is another issue, especially for the flavonoids, antioxidants, and such molecules. And then because the solubility is compromised, so bioavailability is also compromised. Many a times they have poor permeability as well. And the doses which are recommended, or even preclinical data which is generated for these molecules, we talk of doses which is not humanly possible to take. For example, curcumin की कितनी सारी studies हैं जिसमें लोग बात करते हैं 250 mg per kg, 500 mg per kg. So 500 mg per kg को अगर मैं translate करती हूँ to a 60 kg human being, 3 grams, consuming 3 gram curcumin every day may not be very palatable for a person. So if we can in some way come up with a system which could reduce the dose and improve the efficacy of these molecules, that could be an answer to this problem. And several of these molecules undergo first pass metabolism, which also, compromises their bioavailability. So what is the answer? One option I offer is that we could, encapsulate them into a delivery system, which would improve their bioavailability, which would improve their efficacy due to targeted delivery at the site of action, or which could, prolong their shelf life, help in a controlled release, and also reduce any side effects which they may depict. So these are some examples on molecules which we have worked in my lab over the number of years. And the first molecule, which is okay, so before that, so this is a very popular saying by Albert Einstein, that if you judge a fish on its ability to climb a tree, then we could think that the entire life, so the fish may not feel capable. So same goes for the phytochemicals as well. So if they are not performing in the raw form in which they are available, that doesn't mean that they do not work. So it is just that for us scientists and the formulation scientists to show that they can work. So one of the molecule which is very close to my heart is sesamol, which is a small molecule. It fulfills all the Lipinski's rule of 5. So it should be a good molecule to be categorized as a drug. So sesamol is the antioxidant which is not naturally present in sesame seeds. तिल हम सब लोग जानते ही हैं, अभी विंटर्स हैं आजकल नॉर्दर्न इंडिया में all of us are consuming sesame in one form or the other, and in South India sesame oil is really known for its beautiful medicinal properties. Sesamol naturally nahi present hota roasted seeds mein, but you can from sesaminol you can further produce sesamol. So as I said, it's a beautiful molecule with a small molecular weight, but in spite of that still, and as I said, it has a panel of activities, right, from antioxidant, antidepressant, anti-aging, neuroprotective. You talk of an area where they work, as sir was mentioning about cannabis, that it has a variety of receptors on which it acts. And similarly, most of these phytochemicals, antioxidants, they have a profile, because several of these diseases are now related to oxidative stress, inflammation. So, and as a result, since they show a panel of such activities, so they are effective in a variety of situations. So, पहले हम बात करते थे these phytochemicals or natural molecules were referred to as dirty drugs because they showed pluripharmacology and they acted on a multiple of receptors. और अभी जितनी newer lifestyle diseases आ गई हैं, we are now talking of that they have multiple targets, and we need to produce a cascade of effects for a molecule to be effective, and that is what these phytochemicals actually offer to us. So we started some studies way back in 2000 on it as an anti-aging agent, and as I said, in spite of it being a small molecule, it shows a low oral bioavailability, which is not so bad. 35% is good enough for some of the molecules, but it is rapidly eliminated from the body as conjugates. The achieved plasma concentrations are low. Then it is known to cause forced stomach cancers, although humans do not have a forced stomach, but being a phenolic molecule, it could probably cause gastric irritation. So if you want to administer it orally, you need to ensure that it does not cause that acidity. And as it has fast permeation, especially if we want to apply it on the skin, so as a result it is immediately absorbed across the skin, and as a result reaches the bloodstream, and the local action is not achieved. So we prepared sesamol loaded solid lipid nanoparticles. The particle size was around 120 nanometers. We confirmed the stability of the particles. The irritation was reduced, and we would be achieving a local targeting. That was the aim, and the entrapment efficiency and drug assay were good. So why prepare solid lipid nanoparticles? Because most of the molecules, as I said, these molecules have poor bioavailability or poor absorption. So for most biological membranes, lipophilic character is suited, and these SLNs are then coated on the surface with a surfactant or a phospholipid, which further allows them to interact with the biomembranes. If I compare them to liposomes, then they are easier to prepare. The methods which are available are scalable, and the leakiness which is there because the liposomes they have a liquid matrix or a mobile matrix, which leads to leakiness of the entrapped molecules. That is avoided when we are talking of a solid lipid matrix. They are better than polymeric nanoparticles, which also have a solid matrix, because polymers are, one, costly. There could be biocompatibility issues. Even if they form monomers, then those monomers, how they accumulate in the body, could be a concern. And most of these liposomes as well as polymeric nanoparticles require organic solvents for their preparation, while SLNs may require organic solvents, but there are methods available where you can prepare them without the use of organic solvents. So these are a few reasons why SLNs were selected by me. And as I said, so this site shows that the, so this figure, it shows that the amount of sesamol which permeates across skin when it is applied as a free sesamol molecule. So you can see that it is immediately absorbed and permeates across the skin. So it would be lost into systemic circulation. Then we incorporated sesamol into solid lipid nanoparticles. And then since these solid lipid nanoparticles are liquid dispersion, so there could be an issue in their topical application because of their flowability characteristics. So we further incorporated them into a cream or an ointment base. And both sesamol loaded SLNs and their incorporation into cream could reduce the flux across the skin, while when we check the amount of drug which is retained in the skin, and is also shown in the table here, we could enhance the amount which is retained by 300% to almost 400% than when it is applied as a free sesamol. So we developed some models where we evaluated it in skin cancers, and we could show very nice effects with when we incorporated into solid lipid nanoparticles. So this is the study where this is a group, the cancer group, and here we just applied free sesamol, and here we applied. So you can see when sesamol in SLNs was applied, the skin is clear. It is almost similar to the naive control animals. And we compared it with marketed tretinoin cream. The tretinoin cream did cure the cancers or the precancerous lesions, but you can see that the skin is wrinkled, and it is not looking as healthy as the skin which was treated with sesamol loaded SLNs. The reason beyond the sesamol being a good molecule could be because these solid lipid nanoparticles, they are especially suited for skin application because they spread on the skin, and the structure, it collapses, and a continuous layer of lipidic nanoparticles is created on the surface of the skin, which acts as an occlusive and hence hydrates the skin as well. So then we also evaluated it in an animal model of menopause. Menopause is known. We created a surgical menopause model, and menopause is known to cause. Estrogen is a very protective hormone, and which has its own panel of effects, including antioxidant, anti-inflammatory effects, and many a times this stress, menopause related stress, is known to lead to dementia and anxiety. So we took sesamol at doses varying from 1 to 8 milligram per kg, and we administered to these ovrectomized female rats for 7 weeks. And we established by our experiments that these solid lipid nanoparticles, when they were probed by a fluorescent dye, they crossed the blood-brain barrier and reached the brain, as is shown in this slide of brain section under confocal microscopy. And we also concluded that the phosphatidylcholine, which has been used, although at very small amounts in the preparation of these solid lipid nanoparticles, actually enhanced the memory, because there was a significant improvement in the glutathione levels, which are known to be related to the consumption of phosphatidylcholine. And the gastric irritation was also managed by incorporation into solid lipid nanoparticles. Another wonder molecule all of us have, I'm sure, worked with curcumin at one time or the other. It is again, I think it is the small amount of curcumin which is absorbed, and the known bioavailability of curcumin is less than 1%. But somehow that small amount tends to, trigger cascade of events one after the other, which gives very protective effect. We all know it is an antioxidant, anti-inflammatory, analgesic, antimicrobial, wound healing effects, wonderful wound healing effects. Although I'm not showing that data, but we have for the first time reported the anti-biofilm activity of these curcumin when it was incorporated into solid lipid nanoparticles, and also as a cosmeceutical. It is still not classified as a drug because it has a very poor, it is a BCS class 4 type of a molecule with solubility as low as 11 nanograms per ml. It undergoes rapid metabolism. The metabolites, we are still to categorize whether they have activity comparable to curcumin or less than curcumin, or oral bioavailability is less than 1%. And then physiological instability is a big issue with curcumin. It degrades in its formulations in water. Hydrolytic degradation takes place. It is prone to high photodegradation, acid and alkaline degradations, and at most of the pHs, including pH 7.4 and 6.8, it is said to degrade. So this is something which I took up from a cartoon, and it says that I have discovered a herb which can cure so many diseases, but I really don't know how to get it, registered as a drug. So that's one challenge with these natural molecules, including curcumin, that to how to get them into the pool of therapeutics. So the challenge is how to transfer this nascent knowledge from lab to the clinics, and also develop therapeutic strategies for their better availability at the site of action. So curcumin-loaded solid lipid nanoparticles, we've already been granted a patent in India, and we've also filed applications in USA, Canada, and Europe, and we are at a stage where they are being examined in USA and Canada. Europe is a bit slow. So we could improve the solubility by 6 into 10 is per 5 times. Particle size was 200 nanometers, and we could establish 100% photostability of curcumin when it was incorporated into these solid lipid nanoparticles. A zero order controlled release was achieved for 4 days. We established the safety, both oral as well as dermal safety, of curcumin-loaded solid lipid nanoparticles, and also its in vitro cytotoxicity studies. And the beauty of these nanoparticles were that they are autoclavable, and the size, assay, as well as encapsulation efficiency was maintained, and it was also stable under refrigeration for 1 year and up to 6 months at room temperature as well. Most of the nanoparticles, they tend to undergo aggregations when stored at room temperature. The process is scalable. We've already established and demonstrated its scalability with an industrial partner in Himachal at Paonta Sahib. The product was developed using quality by design principles and is optimized. We used high pressure homogenizer for its preparation, which is usually present in most of the pharmaceutical industry. The output process resulted in more than 50 liters per hour. The components which are employed are low cost, easily available, no organic solvent is used, and as I said, the composition is autoclavable, so it can have applications for wound dressings and in infected diabetic foods. So one example of what we did with curcumin SLNs, as I said, we prepared wound dressings. We've combined it with a probiotic for a wound dressing as well. But one example I've taken up here is where we combined it with docetaxel, and we co-encapsulated docetaxel and curcumin into solid lipid nanoparticles, because docetaxel, though a very nice chemotherapeutic agent, is known to undergo PGP efflux, which and curcumin-like molecules are known to offer chemosensitivity or sensitization, where they improve the proneness of the cancer cells to chemotherapy by a variety of mechanisms. Both the drugs, so curcumin has its own plethora of anti-cancer activities already established, though we used it at a very small quantity of around 10 times less than docetaxel, but still they are known to have synergistic actions. The particle size was around 300 nanometers, and we could achieve good entrapment efficiency for docetaxel of more than 80 and around 78 for curcumin in the system. We did a lot of studies, and some of them I'm just exhibiting here, where we evaluated it against 3 breast cancer cell lines: MDA, MB231, which is a triple negative cell line. We took a double negative T47D cell line, and we also took MCF7, which includes estrogen, progesterone, and glucocorticoid receptors. And in all the cases, so we compared free docetaxel with docetaxel encapsulated into nano-SLNs. Then this system where docetaxel and curcumin were co-encapsulated. Then we further, tagged this with a peptide, and we prepared both docetaxel-loaded SLNs with a peptide and the the combination with a peptide. And in all the cases, we could show an improvement in the anti-cancer activity as high as 400 times. And the MICs, the inhibitory concentrations, IC50 values were reduced considerably. This is where we developed breast tumors. You can see big tumors were developed. This is the animals bearing the tumor, and these are the tumors which are isolated from these animals. And when the animals were treated with the B group is only docetaxel loaded SLNs, the tumor size reduced from here to here. So the tumor volume was almost 1000 cubic millimeter for the tumors. When we treated it with docetaxel, it reduced 5 times to 211. And then further, if we treated it with a combination of docetaxel and curcumin, the size reduced further almost 10 times. And you can see there is a minuscule of the tumor, which could probably be a scar tissue was left when the docetaxel plus curcumin loaded nanoparticles with this peptide the animals were treated with this. So we also wanted to give a proof of concept where we used fluorescent labeled nanoparticles. FITC was the dye which was used. And we could establish that the amount of fluorescent dye which enters into these cancer cell lines is significantly increased when they are incorporated into solid lipid nanoparticles. So, and we did DAPI staining to stain the nucleus of these cancer cells. So in case of the free FITC panel, you can see that hardly any fluorescence is observed. When these fluorescent, this dye was incorporated into solid lipid nanoparticles, you can see that there is significant fluorescence in all the 3 cell lines. And when a peptide was also used, the targeting was further improved and there was a significant uptake. So the regulatory status for these nutraceuticals or phytopharmaceuticals is that the public awareness is increasing, as well as public has a great faith in the performance of these molecules. However, the standard guidelines for the production, for the collection of the plants, for the extraction are still missing in most of the countries, though as Dr. Ahmed mentioned that already the efforts have been started at some institutes in India and also by the government of India. So, but there is a flooding of the market by low quality products, especially in developing countries. So there is a need for global regulatory guidelines for protection against spurious and low quality drugs. And there has to be a coordination between formulation scientists and pharmaceutical industry for new relevant products which have extended market and which can actually show efficacy and prove their worth. And the rigorous scientific research, reverse pharmacology needs to be conducted to unravel the mechanisms and potential adverse effects of these molecules. So naturally, so FDA would not let us release them till we prove that they are safe, because efficacy, yes, it has been. So whenever you know the efficacy, so the other concern is safety as well as stability of the product. So we have to establish that. And as I said, they are being widely used. So the right quantity, the correct quality, and the proper regimen needs to be followed. Several of these products are flooded not only in the Indian, but also in the pharmacies and other stores in US as well. So toxicity, as I said, is a concern for most of the nanoparticles, but it is more so for polymeric nanoparticles, inorganic nanoparticles like silver, gold, and other metal nanoparticles. It is not such a big concern with lipidic nanoparticles. That was another reason why I picked up solid lipid nanoparticles. Till date, we've scanning the entire literature, and we've been regularly performing preclinical toxicity studies in my own lab for different lipidic nanoparticles which we have developed, and we've always found them to be safe. There have been 1 or 2 reports, since they are lipidic nanoparticles, where they reported some change in the lipid metabolism, but most of them, they were reverted back in the satellite groups of these toxicity studies. There are, I would say, amongst, if there are 20 studies which are reported, I found 1 or 2 studies where they indicated that there was some change in the cholesterol levels. The concern, as I said, for US FDA for any product which is showing good efficacy is always because that efficacy leads to higher plasma concentration. So safety is always an issue for these. And then some of these phytochemicals have been known to interfere with the gut microbiome. So that could also be a concern for the phytochemicals as well as for the nanoparticulate systems which are developed for these. So as I said, we've done extensive toxicity studies. So one example I show here is for oral toxicity studies of curcumin-loaded solid lipid nanoparticles. We performed acute as well as repeat dose toxicity studies, and we evaluated all the parameters which are listed. And in this case, we did not find any changes in the cholesterol levels. All the liver function tests and the kidney function tests were normal. This is again, and we could go up to a dose of 1,000 milligrams per kg, and there were no effects. So these are some of the achievements. This is my research group, my PhD students and master's students in my lab. And thank you.
Thank you very much, ma'am, for sharing your valuable research, first of all. I always get a new thing from out of it. It's very helpful for me.
Thank you.
And foremost, your words, they always help us to explore the new horizons in this transformative field. And they will surely help students also and teachers also who are doing PhD programs or going for PhDs. Thank you very much, ma'am, for your presence.
Thank you.
And
Thank you
time.
for inviting me.
Thank you very much. Now I would like to introduce our second speaker of the day. Professor Geer Ishaq is a senior faculty member at the Department of Pharmaceutical Sciences, University of Kashmir, with more than 22 years of teaching and research experience. Additionally, he has served as coordinator, UGC Human Resource Development Center, and as director, Center for Career Planning and Counseling of the University of Kashmir. And this capacity, he has organized several faculty development programs, placement drives, and coaching programs also. He has delivered 175 extension lectures outside his department, besides publishing more than 125 popular articles and 52 original research papers in national and international journals of repute. Professor Geer is also a member of several state and national level committees, including advisor, Price Monitoring Resource Unit, member PG Curriculum Committee, Pharmacy Council of India, member coordinator NACP team, nominee CPC SCA, member editorial board of several high impact national and international journals of repute. So I welcome you, sir, and I request you, sir, to please enlighten us on the topic Emerging Frontiers in Pharmaceutical Research and Innovation, Needs and Demands Assessment.
Thank you. Yes, thank you. Thank you very much, Dr. Priyanka Kriplani, for that elaborate introduction. First of all, I would like to express my heartfelt gratitude and thanks to Principal of the Guru Gobind Singh College of Pharmacy, Dr. Kumar Gauravji, other dignitaries on the present at this occasion, then Dr. Geeta Deswal, Dr. Zabir Ahmed, Director IIIM Jammu, and Dr. Indu Kaur and Dr. Ashish Baldi. Many thanks to all of you for having me for this particular session in this national conference. I would just like to share my presentation right away. Please let me know once it becomes visible.
So visible.
Okay. So as mentioned, I'll be talking about emerging frontiers in pharmaceutical research and innovation, needs and demands assessment, keeping in view the theme of the conference. And because, I believe that most of the participants are students. So my intention would be to, acknowledge them and inform them about the roadmap for doing quality and sustainable research. These days, there is a lot of emphasis on conducting sustainable research. Sustainability is the key word. Whichever events we have celebrated over the past 1 year, whether it was the Teachers' Day or any other occasion, the World Pharmacist Day, the word sustainability was inevitably there in the theme. So my emphasis would be on that. And in this conference on advancing pharmaceutical research, methodologies in innovative drug delivery, phytopharmaceuticals, and healthcare policy integration, already some valuable insights have been shared by Dr. Zabir Ahmed sir and Madam Indu Kaur about the phytopharmaceuticals and about how they could be leveraged into the mainstream medical and healthcare sciences. It has been very informative, very insightful deliberation so far. I have attended both of the sessions and have gained several important pieces of information and valuable knowledge. So when we talk about, my intention will be to discuss some basic connotations, basic important aspects of quality and sustainable research with the students. My target audience will be the students in particular. So we are actually going through the Amrit Kal, that is the Azadi Ka Amrit Mahotsav ended in after 75 years of independence, some couple of years back. And we have stepped into the Amrit Kal now. That is, we are all struggling and striving, and we are all working towards achieving our goal of Viksit Bharat by the year 2047, as has been envisaged by our Honorable Prime Minister, Shri Narendra Modi ji. So whatever we do and whatever we plan and whatever we attempt to work at our respective institutions, our goal has to be one, as has been mentioned by Dr. Zabir Saab earlier, how to achieve the Viksit Bharat goal and how to contribute significantly towards this goal. So when we are talking about research and innovation, our strategies and our plans and procedures and our research activities also have to align with this particular goal. That is, whatever we plan today, it should eventually contribute significantly towards achieving this Viksit Bharat by 2047. So in this direction, as shown in the picture in this slide, research and development in pharmaceuticals, in medicines and healthcare is very important, because that will only lead to new molecules, as have been working people, the acclaimed and accomplished people like Dr. Zabir Saab. They have been doing it at reputed institutions like IIIM Jammu or this UI, Institute of Pharmaceutical Sciences, Chandigarh. And simultaneously, we need to work on innovations in medicines. The research has to be innovation-oriented. The research has to be oriented towards bringing new, innovative, novel molecules and making new advances. Recently, this MedTech policy has also been revised, and it has been thrown into public domain for discussion. So MedTech policy is also, focusing on developing these new molecules, innovative molecules, and making our research as innovative and novel as possible, and making it sustainable, which would last for another 40, 50 years at least, and which will bring out results, products, services that will help us achieve our goal of becoming a progressed and developed nation. So let me first discuss what are the various types of research which we can which our students, our scholars can take up, and they are already taking up, so that we achieve this goal of quality research and sustainable research. First and foremost is the discovery research, which is aimed at finding novel interventions, novel targets, what we call as basic research, the basic fundamental research, and includes validation of leads in vivo and in vitro models. This also includes those proposals where proof of concept is already there, but further research is needed. For example, there is a proof of concept of a biomarker or a biomolecule, as extracted from some plant. But now there is, as was being discussed by Professor Indu Ji, that we need to make it, presentable in the form of a formulation, and we need to bring it to the mainstream. Curcumin is already there; it is known for its activities. But how to make it mainstream and how to make it palatable, how to make it presentable, so that it would be efficiently and usefully used by the patients. That is discovery research. That is a part of discovery research. And it also includes drug discovery and new drug delivery systems, new animal models, new tissue-based or culture cell-based models, organ-on-chip, biomaterials, and personalized medicine. And may also include, nowadays, there is a lot of buzz about artificial intelligence, machine learning, virtual reality, and augmented reality. So how we can incorporate those algorithms in our drug delivery and drug discovery process, in our novel drug delivery identification, that is also a part of discovery research. So that is the first type of research. Then second type is the development research. Now, development research is aimed at developing interventions for screening, diagnosis, prevention, treatment of disease, and various other conditions by making existing interventions simpler, safer, more efficacious, and more affordable. The example would be, suppose we are developing a point of care testing equipment or apparatus, or we are developing some new molecular diagnostic test or some new medical device, or any new medical or healthcare technology like artificial intelligence or machine learning based predictive models. That comes under the purview of development research. It could also include clinical trials of vaccines and therapeutics, development of new molecules, new vaccines, new drug delivery systems. That is what we call as development research. Third type is the delivery research, wherein the delivery of these services or health technologies, how to make it better, or implementation of the research which has already been done, the new leads which have been found or identified. This will include health system-based interventions, scaling up of access or implementation of new health technologies. Examples could be how to achieve single-digit neonatal mortality rate in a district. So how will you deliver those services which will reduce the neonatal mortality rate? Or how will you deliver the services which will integrate newer technologies into primary healthcare, for example? Or how would we optimize functioning of our Arogya this Mandir or Ayushman Bharat Yojana, which has been acclaimed and appreciated worldwide in reputed journals like Nature and this British Medical Journal. So how to make our services, delivery mechanisms, distribution mechanisms better and better. That is this kind of research. And fourth and the last one is the description research. Now, description research aims to understand the disease or condition, including its burden, risk factor, and determinants and pathogenesis. And it includes description projects that will be funded if it generates information required for decision-making. Some examples could be like studying the relationship between long-term indoor and outdoor air pollution exposure or pregnancy outcomes, or possible impact of vaccination or pneumococcal diversity. This all includes under description research. So our students could take up any of these types of research, whether it is description research or delivery research or developmental research or discovery research. Any of these research modalities could be undertaken by our scholars. But one thing which needs to be borne in mind while taking up these research activities or research methods is that the research has to be socially relevant and it has to have some impact on the society on the whole. It should not happen that we are doing some research which is devoid of any relevance to the society or which is lacking any kind of impact on the society, whether it is a social impact, economic impact, whether it is any other kind of impact. So some kind of impact has to be there. And that could be made possible by undertaking solution-based research, demand-driven research, context-specific research, translational research, need-based research, innovation-promoting research, research with socio-cultural economic impact, and research which is locally, nationally, and socially relevant. So that is very important aspect. A lot of there is consciousness on these issues. A lot of enlightenment is there these days because, we have been doing research for years, for decades, but we have not been able to achieve the desired results, though now new molecules are coming up even from our country, new leads are being developed, but it took a lot of time. So that's why the focus is more on socially relevant research nowadays. And if we have to measure the research and development output of any institution, that's exactly what we do when we undertake any NAC visit or NIRF accreditation and assessment. How would we measure the research and development output? What are the indicators? The indicators are, as we all know, research publications in the form of papers and books, patents, whether registered, published, or granted, then research projects, whether funded or non-funded, national and international conferences and seminars organized and attended, research scholars enrolled and passed out, research promotion and research ethics, prevention policy, then departments, how many departments are there in the university or the college which are financially assisted by under UGC SAP or CAS or DST FIST, DBT or ICSSR recognition. Then whether we have an innovation incubation, entrepreneurship, startup culture, or a skill development center at the institution, and whether state, national, and international recognition and awards have been sufficiently won by the faculty, and what is the social impact, outcome, and relevance of our research, what we call as valorization of research. Some of the students might not have heard this term, but valorization means what is the outcome of research in terms of products and services, how it is benefiting the society. And that is very important. That is what is gaining prominence these days. So if we see these are the goals which have to be achieved through quality and sustainable research. Now, what are the prerequisites? When we say that needs and demands assessment, what do we need to do? For example, yours is a college which is doing very well in all spheres of academics and research, but you want to proceed further and march ahead. So what are the prerequisites? The prerequisites are, first and foremost, funding, infrastructure, manpower for research, policies and procedures, incentives for doing quality research, collaborations at national and international level, research communications in terms of publications, translational research, integrity in research, and ethics in research. These are some of the prerequisites of doing quality and sustainable research. If you want to measure the quality of your research, you can measure them on these very criteria and indicators. And let us discuss briefly one by one all of them. When it comes to first and foremost, as we say that money makes the mere go. If you want to do quality and sustainable research, which has a good impact on the society, first thing which you need is money. So how will you get the necessary funding, and what are the sources of funding? You can get funding from the state level public institutions like your Department of Health, Department of Higher Education, Department of Science and Technology, or you can get it from private sources like NGOs, like industry, or like public-private partnership sources, wherein the government and non-governmental agencies are tying up, collaborating to fund research. Second source could be the national level funding, which we all know and apply for various projects like ICMR, DBT, DST, UGC, Ayush, etc. They do fund quality research, which is of high impact and high relevance. Then industry, NGOs, and academia. And even at international level, there are a whole lot of sources like World Bank, WHO, UNITAID, United Nations, which fund good quality research, multicentric research, multidisciplinary research, and that can be for sure availed. So once the funding is done, the first step towards doing quality research and sustainable research was to arrange the funding. Now that you have the funding, what will be the next step? Next step will be development of infrastructure. So infrastructure means you should have the research institute or the research center available at your college or the university, wherein you are able to do cutting edge research, quality research of international global standards. For that, you need research equipments, high-end equipment, sophisticated equipments of very good these outputs, which are standardized, validated from time to time. And you need databases, books, and medical journals, which could be referred by the scholars for arriving at research hypotheses, research questions, and for publishing their work. So that is the second most important thing to have for undertaking quality research, that is infrastructure. And once you have the funding, you have the infrastructure, you have the laboratories or research centers, and you have the necessary equipment to do the research, then most important thing to have will be the trained manpower, qualified professional trained manpower who are specialized in those areas where you want to undertake research, who have the necessary wherewithal, training, professional training, who can handle the equipments, who have the skills and competencies, who have the aptitude as well as the attitude to do quality research and to do research of global standards. So then once you have it, you can devise the curriculum and incorporate it into your UG and PG curriculum. Teach them research methodology, teach them research ethics, teach them how to get funding, teach them how to write protocols, and teach them how to publish the scientific research. So that will develop your manpower. So you have the funding, you have the infrastructure, you have the necessary equipment, and you also have the necessary manpower. Now, what will you do next? Next step will be to devise a research policy for your institution. You need to have a proper policy framework, like what to do and what not to do, how to do, when to do, where to do. Only when you have, through research, we generate scientific evidence. That scientific evidence translates into a policy framework. And that policy framework has to be acted upon. It should be followed by action, and that will lead to practice. So it's a circle that is interconnected. That is, research, evidence-based research leads to policy framework, and policy framework, when implemented, leads to practice. So that will fetch you the desired results. So this is the most important thing, that you need to make everything clear in a well-documented fashion. What will be the policy for research at your institution? What will be the do's and don'ts? And what will be the modalities for undertaking research of global quality, of good quality and international standards? Then once you venture into this kind of research, you need to have incentivization. That is, those who are doing well in research and are coming up with good results and output, they need to be incentivized in terms of appointments, in terms of promotions, in terms of financial initiatives or recognition, in terms of fellowships, etc. There's a huge, in our educational institutions, there are not much incentives for doing good quality research. And that's why, there is not, many people don't find motivation enough to do high quality research. So quality has to supersede quantity. Quality has to overcome quantity. That is the first and foremost thing to be borne in mind. And nowadays, we have national education policy in place, which lays a lot of emphasis on collaborative research. So I would like to spend a few moments on this. We often talk about interdisciplinary research, multidisciplinary research, and we treat these terms synonymously. But actually, they are not synonymous. If we look at it from the perspective of social sciences, these are different terms. Whether we call it transdisciplinarity, interdisciplinarity, cross-disciplinary research, pluridisciplinary research, or multidisciplinary research, there is difference among them. They are not completely synonymous or similar. For example, when we talk about transdisciplinarity, this is a state of complete balance of influence. Suppose two departments or two institutions are collaborating with each other, and when there is a balance of influence and there is coordination at the highest possible level, and there is cross contact and cross communication, then we call it transdisciplinary. And the extent and quality of cooperation is so much advanced that this could lead to a new discipline which could be established. If two opposite disciplines are collaborating in a transdisciplinary manner, it could lead to the evolution of a third new discipline. Then interdisciplinarity is weaker than transdisciplinarity because the balance of influence is upheld. However, cross-communication, cross-contact may not be at that level. Then third level is the cross-disciplinarity, where one discipline dominates the other. So in transdisciplinarity, they are balanced. There is no domination. But in cross-disciplinary activities or research, there is domination of one discipline over the other. The dominant discipline is the one which establishes all important premises of that research or that collaboration. At fourth level, we have the pluridisciplinarity, wherein the contact becomes weaker or more sporadic in nature, as was there in the cross-disciplinarity. And a positive factor is that the communication is symmetric. So then we have the lowest level, which is known as multidisciplinarity. This is the least developed form of interdisciplinarity. The communication between various disciplines is reduced to a minimum, but still it is relevant, still it is worthwhile to have, because projects of education or research are often overlapping or complementary to each other, and they would also fetch good results in the long run. So we need contextual and need-based research. We need research which is context-specific, which is relevant to our regional needs, local needs. And we need to communicate this research. So what is research communication? By publishing in it papers, in books, in book chapters, in journals, by presenting it in various conferences, seminars. We communicate. We have to communicate. It should not remain restricted to our library shelves, and it should not, be disconnected from the community or the society in which we live. So that is why communication is important. Having a touch with the society, having a pulse of their problems and issues, so that we devise hypotheses and research problems in accordance with the needs and demands of the society, in accordance with the needs of our community. That's why communication is important, and establishment of entrepreneurship and innovation cells are important. And we can have, in foreign countries, they organize these research fairs and exhibitions wherein they allow community leaders to come in and give their problems, what problems they're facing in their society, in their agriculture, economy, in their polity, in their day-to-day life. Then they devise their research programs accordingly. They come up with solutions to those problems which are being faced by the community, which are being faced by the society. And there has to be integrity in research. We are confronted with a whole lot of issues these days. Plagiarism, which is, taking some paragraph or page or lines from some other source without referring to that source. Or there is duplication. That is, we publish the same paper in multiple journals, or there is gift or ghost authorship. We include authors who have not significantly contributed, intellectually contributed to the development of the paper or in that research, but we give them authorship just because they are doing some plagiarism check, or just because they are doing some other small activity. That is gift or ghost authorship, which is unethical. And then salami publications, you have done a research on a particular plant, let us say, and you have devised some activity, but you're publishing it into bits and pieces in 4 or 5 papers in order to have more number of papers. And that's why I said quality has to overcome the quantity. The quality has to supersede the quality. And lastly, the outsourced publishing. These days, a lot of platforms are there which are offering to publish papers and book chapters on behalf of the scholars. That is unethical. It is not integrity in research. And other ethical issues are there. For example, the confidentiality, privacy, non-maleficence, beneficence, autonomy, and justice. Institutional ethics committee has to be there, which will devise modalities for ethical research. Confidentiality, if you are using human subjects, their privacy has to be maintained. Confidentiality, you don't have to publish their pictures in the journals, in your papers or book chapters, and their names don't have to be revealed. Non-maleficence, you don't have to cause any harm. Primum non nocere was the basic premise of medicine since centuries. First of all, do no harm. So it is good that you are doing some benefit, but first make sure that you are not doing any harm. Then beneficence, then comes the benefit. And autonomy and justice. You have to give the person an autonomy, whether he can stay with the trial or leave the trial. And you have to do justice in case there is any problem or issue, and you have to give compensation and other things. So these were the basic requirements of what are doing quality and sustainable research. But nowadays, one more important thing is that training our PhD scholars to be thinkers. There is a well-known saying by this Gundula Bosch who says that somehow we need to put the philosophy back into the doctorate of philosophy. We are talking about PhD. PhD means doctorate of philosophy, but there is no philosophy inside. So people are not thinking much. Our scholars are becoming so mechanical. They're doing the research in laboratories, they're just jotting down the numbers and reproducing those numbers in their thesis, and then giving results without doing much of thinking. So it is all becoming mechanical, which anybody could do. And for that, you exactly don't need a postgraduate or a doctorate. So that thinking has to be, discussion part has to be the most enriched part of the thesis. Thinking has to be put back into the research, and philosophy has to be put back into the doctorate. Because at the end of the day, as mentioned by Jean Hemestra, at the end of my career, I will not care at all what my H index is, how many papers I have published, or how I will care tremendously whether I was a good mentor, had a positive impact on the lives of others. So what is important? Your name may be figuring in top 10, top 2% scientists of the world. You may have the highest number of citations or the highest number of H index to your credit, but you have not made any significant impact to your community, to your society by virtue of your research. So then at the end of the day, we need to do some soul searching, some introspection, because one needs to care most importantly, one needs to care that one has to be a good mentor and needs to have a positive impact on the lives of others. If you had a positive impact on the lives of others, if your research had a positive impact on the lives of others, your research is successful. Otherwise, it is not, no matter how many citations you have got, how many papers you have published, or how many H index you have, but impact is important. So in this slide, what kind of impact can my research have? It could have social impact, what we call as this impact monitoring, impact measurement, impact assessment is known as valorization of research. So it can have a social benefit in terms of social capital, enriching, adding to the social capital of a nation, or it could have cultural benefits by virtue of adding to the cultural capital of a nation, understanding how we relate to others in the society, contributing to cultural preservation and enrichment, stimulating new approaches to social sciences. And ecological or environmental impact could be measured in terms of impact on natural resource capital of a nation. That is how to reduce waste and pollution, how to take up recycling techniques. Economic benefits similarly could denote contributions towards economic capital of a nation, how to enhance the skill base, improve productivity. And then there could be political, spiritual, technological, legal, behavioral, therapeutic, demographic, ethnic, or humanistic impacts on the society, implications on the society. Unfortunately, as of today, none of the universities in India makes it a compulsory to have one section in the thesis wherein they could say a word about what is the impact, whether it had a political impact or a social impact, cultural, ecological, technological, legal, or any other therapeutic or humanistic impact. That section should be an inevitable part of every thesis. Then only it will become important. Then only scholars will start giving attention towards this aspect of our research, to how to make it socially relevant and how to make it impactful. So there is how to foster interdisciplinarity, how to foster transdisciplinarity. We need to build collaborative teams. We need to have diverse expertise, with clear roles and objectives, with clear assigning of responsibilities. Then we need to establish shared research goals. For example, physics, chemistry, pharmacology, and cognition need to collaborate and devise shared research goals. Similarly, sciences need to collaborate with humanities, commerces, management studies, or other sciences and devise common research goals. Similarly, social science needs to collaborate with sciences. For example, we have this new discipline of social pharmacy or administrative pharmacy these days, which measures the social impact of the research. And we need to then undertake educational and training programs on these transdisciplinary type of research, on joint degree programs or internships and fellowships. We need to utilize advanced tools and technologies like big data, artificial intelligence for these collaborative platforms. We need to encourage open innovation and knowledge exchange through open access journals and databases. We need to promote industry-academia partnership with pharma companies, non-pharma companies, government and regulatory agencies. We need to fund and grant opportunities which promotes interdisciplinary research, multi-partner collaborations. We need to create a culture of interdisciplinary research by incentivizing such collaboration, by developing leadership which is drawn from all disciplines. We need to break down these silos and compartments in which we have been working so far. And we need to dissolve these compartments and do cross communication and do cross-cultural activities and cross-disciplinary studies. And so that, then only the science will become meaningful, research will become meaningful. Because when we look at all aspects of this, for example, you are devising a new plant, you have got a new extract, new biosimilar, bioactive molecule. So when you have to take it to a pharmaceutics person for developing the formulation, then you have to take it to a pharmacologist for measuring its activity. Of course, the basic work will be done by the pharmacognosist. And then, we need to even take it up through with the social science people, humanities people, in order to know what will be the clinical outcomes, humanistic outcomes, quality of life, how it will improve upon the quality of life, and what will be the cost aspects, economic aspects, the management aspects. All these things will become possible only when we do multidisciplinary, interdisciplinary kind of research, collaborative research, as envisaged under National Education Policy 2020. When we break these silos and compartments and we're willing to work together, we are not doing it in our own isolation, in our own cubicles, but we are dissolving those compartments. Lastly, I would like to mention what are the emerging frontiers in pharmaceutical research. Like, we need to identify areas which will foster this innovative kind of research, which will foster this novel kind of research, and which will help us contribute significantly towards Viksit Bharat by the year 2047. So within our own domains, what are the areas? Some of the areas I have identified are pharmacovigilance, materiovigilance, cosmetovigilance, hemovigilance, herbovigilance, and nutraceutical vigilance. So far, we have only been talking about adverse drug reaction monitoring, safety of drugs and pharmaceuticals, which is pharmacovigilance. But we also now have to work and think about the safety of devices, medical devices, which is materiovigilance, the safety of cosmeceuticals, that is cosmetovigilance, the adverse reaction monitoring through blood transfusions and incompatibilities, that is hemovigilance, and a lot of work going on pharmaceuticals. So herbovigilance, we should not be bogged by the myth that these herbs are totally devoid of any side effects. That's not the case. Every system of medicine coming under allopathy or Ayush has some certain side effects which need to be looked into. And nutraceutical vigilance, earlier it was mentioned by Professor Indu Kaur ji that this Hercules, father of medicine, has said several centuries ago that make a food thy medicine, make medicine thy food. So that makes it important to do nutraceutical vigilance. Then another area is personalized and precision medicine, and holistic, integrative, and lifestyle medicine. See, our pharmacists have simply been dispensing drugs for centuries together. That is the only job which they have been doing, dispensing medications along with some amount of patient counseling. But I think now they have to switch over, and they have to step into other domains also. How to prevent disease. Only curative part is not sufficient. The preventive, proactive, promotive healthcare, rehabilitative healthcare is also important. So our pharmacists need to venture into these areas which promote preventive healthcare, promotive healthcare, proactive healthcare, rehabilitative healthcare through holistic, integrative, and lifestyle medicine. So they need to venture into this. They need to have this in the curriculum. They need to get trained into this and come up with new research ideas in these areas, because a good number of diseases are just lifestyle diseases, chronic diseases in particular, like diabetes, thyroid, or these PCOS disorders. So they can be rectified through lifestyle modification. Our pharmacists need to know that, and they need to, they need to give that kind of information to the patients. Then there is telepharmacy and bioelectric medicine. There is this upcoming field of pharmacogenomics and metabonomics. Omics studies is going to be the future. Gene editing, this technology, CRISPR technology, that is the future, wherein a lot of playing will be there with the genes of a person. So all these techniques, whether it is the pharmacogenomics or transcriptomics or proteomics or pharmacometabonomics, they are going to play a very essential, important role in coming years. So there are a lot of novel research can be possible in this area. Then areas like pharmacoeconomics and health outcomes research, very important for maintaining equity, parity in health outcomes and maintaining affordable, accessible healthcare in our society. Then there is pharmacoinformatics and pharmacometrics, like we had the bioinformatics earlier. But pharmacoinformatics is also going to play a key role in future. Pharmacometrics is going to play a very key role in future, because you cannot put populations like pregnant ladies or small children to clinical trials all the time. So you need to devise measures how to assess and evaluate their activity, their dosages, and their adverse reactions based on certain algorithms. Artificial intelligence and machine learning, already our students have started, using ChatGPT for writing papers, for writing book chapters, for doing many activities, but there is a limit. We cannot submit a paper which is AI generated. but we can make good use of these technologies. It's not that we should tell them to stay away from artificial intelligence. Many people say these days that we are telling our children to stay away from gadgets, but that is not the solution, because we need to tell them how to make the best use of the gadgets, how to make the best use of the technology, rather than staying away from the technology, or rather than staying, abandoning this new upcoming. We cannot abandon. This is the future. This is the reality of our times. We need to tell them how to make the best use of these technologies. Then there is the area of nanomedicine and nanodiagnostics, big data analytics, clinical data management, 3D printing and robotics, cell therapy, genetic engineering. There is the area of functional medicine, translational and regenerative medicine, clinical research organizations, knowledge process outsourcing, biosimilars and biopharmaceuticals. All these areas comprise of new innovative areas where a lot of research can be done, which can be quality research, sustainable research. So I had added one slide or two slides each in each area, but I don't think there will be sufficient time to discuss these slides in more detail. So these, I have discussed quickly, but I have also tried to devise a research problem in each area. So I'll just restrict myself to what research question could be the innovative research in this area. For example, the personalized medicine and genomics. In this, the research question could be, how can genomic data be integrated with real-time clinical data for personalized treatment plans? And what new biomarkers can be identified for more precise drug targeting? Some of our scholars can work in this area. Similarly, in case of a role of artificial intelligence and machine learning, the research question could be, how can artificial intelligence models be trained to predict drug toxicity, efficacy, and optimal dosing regimens? What role can AI and machine learning play in repurposing of drugs and drug delivery systems? Nowadays, a lot of buzz is there about repurposing. Old molecules which are no more in use are being repurposed for new indications, for new therapeutics. So that can be worked, using AI algorithms. We can determine their efficacy in treatment of these conditions. Then there is nanomedicine. So we can have a research question. How can nano carriers be engineered to enhance drug penetration in difficult to reach tissues like brain, tumors? What are the long-term safety and toxicity implications of these nanoparticles? Would be the possible research question. In case of immunotherapy and vaccine development, the research question could be, how can mRNA technology be applied to other diseases beyond COVID-19? What innovative drug delivery systems can enhance the efficacy of immunotherapies? Similarly, in case of regenerative medicine, the research question could be, how can stem cells be manipulated to differentiate into desired tissue types for drug testing and therapeutic applications? And what are the regulatory challenges in developing stem cell-based therapies? In case of microbiome and gut pharma interactions, how do microbiome alterations affect drug metabolism and therapeutic outcomes? What interventions can be made to modify the microbiome for improved drug responses? In case of advanced drug delivery systems, how can these drug delivery systems be designed for controlled release and targeted delivery in specific organs or tissues? And what are the challenges in developing multimodal drug therapy systems which combine imaging agents and diagnostic tools? We have these techniques of nanosensors and nano delivery technologies. How can they be leveraged to deliver these drugs? Similarly, in case of biopharmaceuticals and biologicals, what are the most effective strategies for enhancing the stability and shelf life of biologicals? How can we optimize the delivery of biological agents, particularly for conditions like cancer and autoimmune diseases? And then there could be research questions for sustainable drug delivery and green chemistry. And lastly, if we submit these projects to ICMR, for the sake of information of our students, I have 2 slides left. So I would just try to inform them, how does the agencies, funding agencies like ICMR evaluate their research protocols? So this is how they evaluate it. Out of a score of 100, 20 marks are given to your background and rationale of the project, which is very important. How is it likely to solve a particular priority problem? Then another 20 marks are given to possible impact. We discussed a lot about social relevance and impact of research. Nowadays, a lot of thrust is given by funding agencies also. When they fund certain research projects, they give 20 marks to possible impact. Is it likely to have an impact on health outcomes? 20 marks will be allotted to that. Then novelty, innovation, whether there is anything new, whether your project is going to add anything new to the existing body of literature is very important. So is the study developing or testing any new idea, whether there is anything innovative or novel in it? 15 marks will be given to that. Then your research methodology is very important, whether you're making use of new methods, innovative methods, you're not using old methods only, but you are using upcoming new methods to achieve the desired objectives, 30 marks. And what is your implementation strategy and milestone? Is the study feasible in a timely manner? How will you get the desired results or outcomes? What is the strategy for that? So 15 marks. So out of 100, this is the distribution of marks. And if you look at the protocol which you have to submit to Indian Council of Medical Research, you have to give clear idea, clear elaboration, details about how does your area cover a particular area. What is the problem statement? What the research question is the most important part of a protocol. What is the problem statement? Where does the problem lie? Where there is a gap or deficiency in the literature, you have to identify the gap in the literature. Then you have to give the rationale of the study, the need for the study, and how your study proposal is going to add new knowledge to the existing body of literature, or how it is going to add new technology or new capacity or new competence. And then hypothesis in 100 words, study objectives have to be very crisp, very brief, to the point, and they have to exactly address the problem. And finally, the methods. Methods should be in complete sync and alignment with the objectives. Whatever objectives you have studied, the methods should be devised to address those particular objectives only, not beyond that. The implementation strategy, expected outcomes, what are the deliverables from the project, what is the immediate next step following the end of the project, how will it, how will you translate it into some kind of product or service to the patient. So translational research becomes important, whether the study is going to generate new intellectual property in the form of patents or not. And lastly, the references. So these are some of the important points for making our research of high quality, global college standards, cutting edge research, and making it sustainable for future, something which will significantly contribute towards Viksit Bharat. So let us all take a pledge to further Atmanirbhar Bharat and Viksit Bharat by the year 2047 through our research, through our academics, through everything that we do. That's my sincere request to all the listeners. Thank you very much for your patient audience. Over to you, Dr. Priyanka.
Thank you very much, sir, for sharing your immense knowledge regarding various types of research and the importance of collaborating with various fields. Like said, in pharmacy, we should collaborate with all. Pharmacology should come together, pharmaceutics, pharmaceutical chemistry, and definitely it will give us a fruitful research which may be beneficial to the society, or it may help to solve some of the environmental problems, or maybe cultural. And if we talk about technology also, like as you said, and definitely we will collaborate in our institute first.
Right. Thank you.
While preparing a research proposal. Thank you very much.
That is the need of the hour. All of us have to collaborate, dissolve the silos and compartments, and we have to come up with innovative solutions which are collaborative and collective in nature. Thank you for your patient listening.
And importantly, sir, AI, the use of AI,
Yes,
that
absolutely.
most of us are doing, maybe just to create whole of the research paper, and we are not using our minds. We have stopped using, like when we started using this mobile phones. Earlier, we used to remember all the numbers. Now we are dependent on this.
Absolutely.
We want to see a number, we have to see the mobile. What is the number of
So,
that
yeah,
friend?
yes, as I mentioned, it is not a bad thing per se, but it can show us the way. We can use it constructively, progressively, and productively. And we can take the leads from this artificial intelligence and chat GPT. Once it lays the foundation, what is the roadmap, then we can work on it. So we need to teach our students how to make the best use of it without entirely depending on it and without using it wholly and entirely. But thinking, as I said in one of my slides, thinking is going away. We should not let the thinking part to go away. Using our minds and mindsets is very important, and incorporating thinking and philosophy into our research is very important.
Thank you. Thank you very much, sir, for your time and presence, most important.
Thank you very much for inviting me. Thanks a lot. It was a pleasure.
Thank you so much, sir.
Thank you, Dr. Geeta ma'am.
Now, I would like to introduce our next speaker of the day. That is Dr. Ashish Baldi, Professor, Department of Pharmaceutical Sciences and Technology, MRS, Punjab Technical University, Bhatinda. He is recipient of highest acclaim to a teacher by Government of India, that is Rashtriya Shikshak Puraskar by Honorable President of India, and APTI Pharmacy Teacher of Eminence Award. He is consecutively featuring among the top 2% scientists across the world in the field of pharmacy and pharmacology for last 6 years by Stanford University, USA, and elsewhere. And with over 21 years of experience, 7 patents, 9 other IPRs, 12 books, 4 special issues, and over 160 national and international publications, more than 40 book chapters, and over 75 invited lectures, several popular articles, more than 175 papers at conferences, seminars in India and abroad, and several best paper awards to his credit. He is an active researcher in various facets of pharmaceutical sciences and biotechnology. He has completed 15 research projects granted by various government agencies like Ministry of Ayush, DST, ICSSR, ICMR, FITM, PSCST, AICT, etc., and transferred to technologies to leading pharmaceutical industries. He is also chief mentor of BCL AICT Idea Lab and received a grant of 1.11 crores for this. I request you, sir, and I request you to please share your knowledge about the QBD state pharmaceutical products, practical perspectives.
Thank you so much, ma'am, and thank you so much, Dr. Geeta and Dr. Kumar Gaurav and his team for inviting me to this wonderful conference. Since morning, I have been connected with this particular event, and once I thought that the concept or the title or the theme of the event is very opt, but after listening Professor Geer and Ishaq sir and Professor Zabir sir and then Indupal Kaur ma'am, I must say that when Indupal Kaur ma'am has started, it went to molecular level and preclinical and clinical and all those pharmaceutical product development. When it went to Professor Zabir Ahmed sir, it was an example, leading example of how a concrete effort can lead you to the best of the outcomes. And then we came to Professor Ishaq sir's lecture, and I found it Gagar Mein Sagar, because it was very necessary when being a academician and researcher, we go deep into our own subject, but we generally lost away the holistic scenario of the research. And Professor Ishaq has very appreciably delivered various aspects in holistic nature, that how a research and research environment can contribute to a particular research problem. And overall, I think this is very apt for the theme. Now I am starting my lecture on quality by design. And this quality by design, as we all know, that we have talked about, product development for herbal or phytopharmaceuticals. We have talked about the success of pharmaceutical industry as such. But when we talk about the success of pharmaceutical industry and its ability to develop product, I think quality by design is one of the key principles involved in the successful product development in pharmaceutical sciences. And therefore, we have to see the practical perspective, how a product develop and how we can ensure the quality of the product when we talk about a pharmaceutical product. So the learning objective of today's lecture will be to understand the process of quality in pharmaceutical products, to identify the process variables in pharmaceutical product development, whether it is phytopharma or whether it is normal pharmaceutical product, to perform critical alignment study and quality risk management metrics based processes, and then to develop process by quality by design strategy. So when we go ahead with this, the content of the lecture will be its background and significance of QBD, then an introduction. Then we have to go in deep into the quality by design principles, and we have to see what are the critical variables, quality risk management, how we can develop a product based on quality by design, and then what outcomes we can get. But whenever I teach in the class, or whenever I take a lecture, I associate this particular scientific principles with the life, and the outcome of those principles with the life should also be learned, so that the student and the learner should identify the relevance of that particular technology in the life. So when we talk about quality by design, everybody of us goes to Starbucks, goes to cafe coffee day. But what is astonishing is that in home, if we prepare a coffee every day, the taste of the coffee is different. But when we go to cafe coffee day, or when we go to Starbucks or any leading brand, the quality of the coffee is same. Now, how it is possible that every time at various places a product has been prepared and the quality remains the same? That is, I think, the best example of quality by design practices in routine life or daily life. And therefore, we have to understand that if we optimize a particular process, that only is not necessary. We have to see the quality perspective of the product development, and then only we can go for the quest of perfection. When we talk about perfection, it's an example that if you try to make an earthen pot from the soil, you have to learn so many things. You have to learn the technique, but more than that, you have to learn how to prepare and how to do it and what not to do it. And therefore, it is always essential that when you, as Ishaq sir has also said, that quality should be preferred over quantity. So here also, when you talk about product development, it is, the quality of the product should be ensured in such a way that you are developing it every time with the same quality. So quality, as a statement said, that quality can't be tested in the outcomes, but it has to be built in. So whether it is your personal quality, whether it is your personal performance, whether it is your professional performance, you have to inculcate the quality within your efforts, and then only you can achieve a specific target or achieve a specific aim in your life. And that perspective of putting quality into your practices comes from your own learning, your own experiences, your capabilities, and also skills. And then your competency, and repeatedly you are trained on something, then only you can gain the knowledge of putting quality into your practice. And that's where the concept of quality by design comes into the life, or comes into the pharmaceutical product development. So when we talk about quality, there are five major definitive parameters. One is, we have to define quality first. Then we have to measure the quality parameters, and then we have to analyze that what is the impact of either process parameters or variables on the quality. Then we have to understand how we can improve and ensure the quality. And then after all that, we have to ensure our practices, that how we can control a particular quality in desired manner. So that is where that 5 steps of quality defining processes comes into the picture. So what are the two approaches? As in our routine life also, either we learn from mistakes, and that was the traditional method of quality control, that a target is defined, and if product quality variation is there, we reprocess, rework, and then improve the quality, and that is called as quality by quality control method. And this was the earlier method used for optimization and production of pharmaceutical products. So in simpler senses, here it is by learning from our own mistakes, learning from failures. And when we go for quality by design or Six Sigma way, it is basically like learning from experiences and giving an outlook that how we can produce a product with consistent quality, how we can reduce the wastages, and how we can increase the productivity with desired quality of the product. That is quality by design. So we will go deep into the quality by design practices in pharmaceutical product development. And before going into it, there is a guideline by International Conference of Harmonization, that is Q2 guideline, which defines quality by design. It's a systematic approach to develop a particular product that begins with predefined objectives. Our target profiles should be predefined, and then emphasize on product and process understanding. That means the raw materials and the process parameters, and then on process control. That is the quality control and assurance part of the product development strategy development based on sound science and QRM, that is quality risk management. So this is where principle of QBD involves: one is systematic approach to process development, that means systematic understanding of the product development process. Then risk and knowledge based decision based on our prior experience, based on our own repeated practice of that product development. We can identify the risk associated with various elements, and then we can take an informative decision based on our knowledge for that. We can continuously improve by experiences, and then leading to develop capable process which can give us the quality product. So what are the steps? One is target quality product profiling, that means TQPP, where we have an intent to develop a particular pharmaceutical product, whether it is phytopharmaceutical or other way around. We have to identify the active pharmaceutical ingredients chemically and physically, and then we have to identify the target quality parameters which we are looking for in the developed product. And then after that, we have to understand the process in terms of manufacturing process, in terms of formulation design process, and we have to understand the design selection of the process part. And then we have to identify how we can control these attributes, whether it is material attributes, whether it is process attributes, in order to ascertain or fix the definite quality attributes that are required for defining the quality. And then how we will measure and ensure that repeated quality is produced. So these four are the major stages of QBD process development. And we talk about success in product development, or we talk about success in life. We have to understand whole of the process. We have to understand whole of the product, whole of the element. And whenever I must say that whenever you are in a difficult situation, you have to assess the situation nicely. What are the causative factors? What might be the effect of this? What are the environmental factors or associated factors? And if you identify those factors, you can easily come out from that particular problem or with a success. And therefore, quality by design principle have its own methodology to assess the impact of various variables, and that is known as Ishikawa fishbone diagram, where we generally take care of five variables, 5 M's: men, that is manpower, their training, their skills, and their accuracy; materials which we are using, their quality, and also the impact on the quality; the methods we are using in terms of process parameters; and then machines which we are using in terms of their performance; and then milieu, the environmental conditions. All this has to be controlled in order to get a product with targeted product profile. And that particular thing we can do with the Ishikawa fishbone diagram to understand the relation between the quality of the product with respect to quality product profile, TQPP, and then the process variables. So what are the steps as far as quality by design based product development is concerned? Firstly, we have to define quality target product profile, then quality attributes, and then process parameters. In terms of materials, we can define CMA, quality critical material attributes. Then we have to prioritize CPP and CQA, that is critical process parameters and quality attributes, and we have to understand the risk involved in terms of risk assessment and management, how we can manage that particular risk in the product development part, and then we can apply formulation by design and experimentation and validation within the given design space. And then we can think of pilot scale production and commercialization with the scale up, successful scale up technologies. So when we define and control everything related to the product development, then only we can successfully transfer it to the commercial product. And that's why it is very essential to identify these three terms. Number one, critical process parameters, that are various processes, pharmaceutical manufacturing processes are involved in one or the other product development processes. And then those processes, we have to understand what are the parameters. For example, if we are doing mixing, what is the mixer speed, what is the mixing time for that? These are the process parameters, and out of those we have to identify that what are the critical process parameters which are critically affecting the quality. Similarly, material attributes, we are using raw materials, we are using API, so we have to understand the physical, chemical, biological and microbiological property of those materials and understand the interaction between those in order to define the product in terms of quality. And then we have to understand their relation of process parameter and material attributes with the critical quality attributes, and we have to identify what are the quality attributes they are impacting and how they can impact. So once we have the quality target product profile in mind, we have to correlate it with the process parameters and material attributes, and we have to critically assess that how we can define the critical materials, critical process parameters, and critical quality attributes. So we have to see the potential impact on safety and efficacy for that particular material or that particular process parameter. And if it is critical, it will be known as a critical process parameter or critical material attribute. If it is not critical, depending on the severity scale, we can assess whether it is a critical parameter or not. Once the parameters criticality is defined, we can define critical parameters, critical materials. In relation to that, we can also define the critical quality attributes which are responsible for performance of a drug product. So these all are based on the risk associated. Whether the risk is high, it is more critical. If the risk is low, it is less critical. So there are guidelines available for adoption of these product development practices. ICHQ8, that is pharmaceutical product development guideline is there. ICHQ9 is quality risk management, and ICHQ10, that is pharmaceutical quality system, PQS guidelines are there for the product development. And if we implement those guidelines religiously into the product development, every time we can ensure the quality of the product is same. So what is quality risk management? Quality risk management is nothing but we have to identify the potential factors or variables which are affecting the quality, which are a risk for the quality. And then if we identify those processes or those parameters or those variables, then we can think of managing them with in order to get the desired outcomes. And Warren Buffett has said, I have simplified it in Hindi, दैट रिस्क तब आती है जब आपको पता नहीं होता कि आप क्या कर रहे हैं. तो रिस्क को असेस करना in terms of what processes you are using, what product you are developing, that is quality risk management. And this is defined by Q9 guideline of the ICH. where we define the risk associated with the, first we identify the risk, analyze the risk, evaluate the risk, and then control strategy for risk reduction and acceptance. How we can continually improve the product in terms of quality is the fundamental outcome of these particular practices. So we one of the method is we can calculate the severity score, and based on that, risk assessment matrix is formed where we can define a particular product risk likelihood on the consequences of quality of the product as high, low, or medium. And then but all these processes and optimization and quality control strategies are defined to the particular design space, the limits where we are talking about, the limits where we are talking about or doing the optimization. There only we can give certain values for optimization, and therefore it is always said कि अपनी हद में रहोगे तो सफलता आपके कदम चूमेगी. That means you have to be in limits. Whenever you have to perform certain, you have to achieve certain goals, you have to be in limits, you have to perform at your best. And if within that limit, that is design space, in terms of variables, in terms of material concentration, or in terms of process parameters, you can achieve the desired output in terms of quality parameters. So how we can go ahead for QBD based development? One is to define parameters, that is quality parameters, process parameters, and materials. Then you have to identify the process, that what formulation process we will be going to implement. You have to understand the impact of the material and process parameters on the quality. And then you have to identify the critical variability, chances of variability, or we can say risk. And then you have to implement the risk control measures and continuously monitor it for the quality. So there are two phases: product and process development and understanding, and then risk control and assessment strategies. So this is the structure of QBD based development in any of the formulation you are developing. But more than the formulation, if you go in life, whenever you face a problem, you must have to understand that what situation has been arised because of this, that problem is coming in our life. Or I can say that what what are the factors contributing to that particular problem. And if we identify those factors, immediately we can identify the impact of those factors on our own life. And in that way, we can learn that what are the aspects of these, what are the what are the relation between these parameters, these persons in our life or in our problem, how they are contributing or resolving, how we can manage them, and how we can get our desired result in terms of what we want to get. So basically, quality by design is not only a pharmaceutical practice, but if you understand the particular process of achieving something, achieving equality in certain outputs or certain outcomes in life, whether it is pharmaceutical development process, whether it is your own personal aims and goals, if you identify those parameters, I think you can best come up with a solution where you can control your life in a better way or control your product in a better way. So from this brief lecture, we have discussed that how we can identify material attributes, process parameters, and quality attributes in every process of life, whether it is pharmaceutical or not. Then for desired outcomes, we have to find out their correlations. And then in order to understand any pharmaceutical process, we should understand the ingredient, process, and quality characteristic and their relation in between, so that we can identify and develop a particular product of desired output. So one is the pharmaceutical learning, that implementing quality by design practice can be a proper solution for development of phytopharmaceutical based product. But the other way around, learning for life is that सिर्फ प्रयास ही काफी नहीं, हर गलती से सीखना भी जरूरी है. सफलता की निरंतरता के लिए प्रक्रिया में गुणवत्ता भी जरूरी है. That means if your attempts only is not necessary, but your ability to learn from those mistakes, converting it into the skill or experience, and then out of that inculcating the quality practices in your efforts will lead you to the success in pharmaceutical product development and in life also. So after going all this, there are certain aspects which our lab is working, and as Ishaq sir has told, that your social relevance, technological relevance, and your contribution towards research should also be identified. So we are working on total quality management or quality based product, quality by design based product development. Two of the products has been transferred to pharmaceutical industries. 16 products have been transferred to a herbal industry, and then there are certain 100 plus 780 publications has been, and 15 grants, major and minor, has been received in this particular area. But more importantly, we have contributed to societal and regulatory contributions also. We have incorporated some suggestions in the national guidelines for probiotics, and then we have also suggested breast cancer screening guidelines to ICMR 2 years ago, and then they are working on it. And we have also worked on good agriculture and collection practices for the local residents of Punjab in order to develop their financial sustainability. And we have 7 GSCP developed, 13 monographs, and 13 SQC documents we have developed in herbal pharmaceuticals. Then we have implemented artificial intelligence based innovation. A mobile app has been developed, that is QCheck, for quality checking of the herbal entity. And what we are doing is we are identifying a herbal drug just by a mobile application. You take a picture of the herbal drug and you can identify not only the drug, but also if there is any adulteration into that. And the other aspect is biotechnology, where we are working on some biofertilizer development, and these all projects are granted by various funding agencies, including Ministry of Ayush, ICMR, DBT, DST, and all other major leading government organization. So with this, I would like to thank everybody for this wonderful conference. And I especially thank the team of Guru Govind Singh College and the Bela College, Professor Shailesh Sharma, a very good friend of mine, Dr. Kumar Gaurav, and Dr. Shailesh, and the rising stars, as I always say, Dr. Geeta, Dr. Priyanka, and the team associated. So thank you very much for giving me an opportunity. Thank you. Over
Thank
to
you very much, sir, for sharing your valuable research insights. And we deeply appreciate the time and effort you spared to enrich our knowledge about QBD, its principles, stages, and structure. And most importantly, your motivating words that we should learn from our mistakes. We should not feel demotivated, or else we should learn it. And again, we should move on towards the success. Thank you very much, sir.
Thank you. Thank you, ma'am.
And most importantly, I want to thank all the speakers and our guest of honor, chief guest of the day, and all the members who participated in the conference to make it a successful conference. Like all things end, so has been this conference. And we witnessed here the remarkable ideas, insightful discussions, and inspiring takeaways. So this conference has been a testament in this field. And on the behalf of organizers, also an association of registered pharmacists, I extend my sincere thanks to everyone who contributed to towards its success. So thank you very much, all of you. Thank you very much. Okay. Shailesh sir, please sir, आप vote of thanks दीजिए if you are available.
So I'm thankful to all the participants who gave their precious time for the successful of this event. And unse zyada thank you very much for the organizers of the conference, Dr. Kumar Gaurav sir and CEO of Guru GGS Yamunanagar. And we are very thankful, give sincere thanks and remarkable note for our today's chief guest from IIM Jammu, Zakir sir, and guest of honor, Dr. Deependra Singh sir, who gave their precious and valuable time for us. We are thankful to all our eminent speakers, Indupal ma'am, Ashish Baldi sir, for their valuable, and Geer Ishaq sir for their valuable contribution for the successful of this event. Last but not least, I'm thankful to the Geeta madam and Priyanka madam who well organized this conference to all of us. So thank you very much. Vande Bharat.